Hi everyone. Today's presentation will be on can CRISPR gene editing cure inherited diseases? Next slide. So what actually is CRISPR? Well, CRISPR stands for clustered regularly interspaced short palindromic repeats. While it is now associated with the world of gene editing, it was actually based on bacteria's natural immune defense against bacteriophages, viruses that attack and replicate within bacteria. Essentially, when infected by a virus, a bacterium will kind of incorporate a part of the viral DNA in its own genome. These spacer sequences are stored in what's called a CRISPR array. This helps the bacteria remember the virus, kind of like memory cells in the human immune system. If the same virus attacks again, the bacterium transcribes the stored viral DNA into RNA, sort of like protein synthesis. This guide RNA binds to a Cas9 enzyme, and once this gRNA Cas9 complex finds matching DNA in the new virus, it cuts it, effectively neutralizing the virus. Researchers took inspiration from this mechanism and developed CRISPR-Cas9 gene editing technology. The procedure in humans is pretty similar, but it has some fundamental differences. First, scientists identify a sequence of the genome causing a health problem. For example, a mutation in the CFTR gene causing cystic fibrosis. Then, they create a guide RNA, gRNA, complementary to that faulty DNA sequence. Because unlike in bacteria, humans don't create gRNA to combat problems in DNA. This gRNA binds to the DNA sequence that it's complementary to it. That it's complementary to. Next, the guide RNA directs the Cas9 enzyme towards it and the target DNA. The enzyme then cuts the DNA at the targeted location, making a double-stranded break. The cell tries to repair the break, and in doing this, scientists can either A, allow the cell to repair the DNA on its own, but this often leads in mutations, disrupting the faulty gene. Or scientists can provide a new piece of DNA that the cell can incorporate during the repair, effectively editing the genome. This makes it useful in gene therapies to treat genetic diseases caused by a known mutation by disabling or correcting faulty genes causing the disease.
So we wanted to talk to you a little bit about the different applications of CRISPR, especially in not just treating humans with inherited diseases, but also plants and other animals to see the really wide variety of applications. So the first thing I'm going to talk about is beta thalassemia. So essentially, this is where the beta globin chains of hemoglobin are either partially produced or they're not produced at all. So the significance of this is in a hemoglobin molecule, you'll have different chains. And in hemoglobin A, the hemoglobin is made up of 2 beta chains and 2 alpha chains, as you can see in the image. So if we're not having enough production of beta globin chains, then hemoglobin A production is affected. Hemoglobin A is found in adults, and it's caused by a point mutation, which is essentially where a single nucleotide in DNA is replaced by another. So this happens in chromosome 11 in the beta globin gene. And this is an autosomal recessive disease, which basically means that if you have one from each parent of this mutated gene, you would get beta thalassemia. If you only have one copy, then that's beta thalassemia minor, which is usually asymptomatic. But if you have 2 mutated genes, then this could lead to either beta thalassemia intermedia or major. So intermedia is when you have 2 mutated genes, but that's for reduced production, not like completely banning the production. So with reduced production, you'll have the intermedia version, whereas you'd get beta thalassemia major if you inherited 2 mutated genes for the complete absence of production of beta globin chains. So if you look at the picture, because there's 2 beta chains and 2 alpha chains, if the beta chains are not produced at all, then you're going to have a lot of alpha chains floating around in the red blood cells, and that causes hemolysis. This can happen either in the bone marrow or by the spleen's macrophages, and that causes hemoglobin to spill into the plasma, where the heme is recycled into iron and bilirubin. Now, bilirubin is the buildup of that would cause jaundice, which is one of the symptoms of beta thalassemia. Hemolysis can also cause hypoxia. So the significance of this disease and why we really need to treat it is because with the major thalassemia, it can be life-threatening within the first 2 years where you develop anemia. Other symptoms include a swollen abdomen, delayed growth, and deformities in the face and limbs, and that's because the bone marrows are growing a lot more, so they're extended to try and replace all this blood that's been like not effective. So the typical cure for this would be transfusions to try and replace the faulty blood cells, the faulty hemoglobin, to try and make up for it. So we would instead try and use gene editing to increase fetal hemoglobin production because fetal hemoglobin doesn't require beta chains. And so Vertex Pharmaceuticals and CRISPR Therapeutics, Casgevy, utilizes CRISPR-Cas9 technology to edit a patient's hematopoietic stem cells. And that editing process aims to increase the production of fetal hemoglobin, compensating for the defective adult hemoglobin. These therapies offer the possibility of a one-time curative treatment, reducing or eliminating the need for lifelong transfusions and associated complications. So the statistics for this were that 25 out of 27 patients who initially needed transfusion because of this disease then became independent of transfusion. But despite the promising results, challenges remain, including the high cost of treatment and accessibility in low resource settings. But this is something that usually gets better as the treatments become more widely available. So the next thing to talk about is rice blast, which is a plant disease, and it is caused by a fungus called Magna Porthae oryzae, which attacks and destroys rice plants, causing significant yield losses worldwide. And by significant, I mean 10 to 30% yield losses annually, and that's enough to feed 60 million people for a year. So this is a very significant disease, which would be great to treat. And the problem is that many rice varieties are susceptible to this. So scientists use CRISPR to knock out or modify susceptibility genes in rice plants, such as the OSERF922 gene. By editing these genes, the plants gain resistance to the fungus. So because they showed such strong resistance due to this CRISPR editing, it was really good to cure the disease, effectively curing the inherited vulnerability, especially without affecting the yield. So I just wanted to show that it's not only humans, but plant inherited diseases can also be cured. And in this case, it ultimately benefits humans too. And the next thing here is Duchenne muscular dystrophy, which is basically caused by mutations in the dystrophin gene and causes muscle degeneration. So researchers used CRISPR-Cas9 to target and correct the mutation in the dystrophin gene in mouse embryos and mouse muscle cells. And the treated mice showed restored dystrophin production, improved muscle function, and delayed disease progression. So there is potential to treat inherited diseases of humans, plants, and other animals using CRISPR technology.
So here are some advantages or benefits of the use of CRISPR to cure inherited diseases. CRISPR technology allows scientists to precisely target and modify genes with greater ease, speed, and cost effectiveness compared to previous gene editing techniques. The gene editing technology can be used to edit mutations in human cells and treat genetic disorders, including sickle cell anemia and muscular dystrophy, as previously discussed. Studies have also shown that infectious diseases and some forms of cancer may also be able to be treated using CRISPR technology. Precision. CRISPR-Cas9 has highly precise targeting capabilities, allowing specific modifications to be made. Off-target effects occur when CRISPR edits unintended DNA sites, which lead to potentially harmful mutations. The guide RNA, however, is carefully designed to only match the target sequence. As well as this, high-fidelity Cas9 variants can be used, which increase specificity by requiring a more exact match between the guide RNA and the target DNA. This targeting of specific regions of the genome minimizes the risk of off-target effects and unintended consequences, increasing the likelihood of a desired outcome. Long-lasting effects. Epigenetic modifications use a Cas9 version, which is dead, which is fused to the epigenetic modifiers such as DNA methyltransferases or histone acetylases or deacetylases. Epigenetic modifications mean that the DNA sequence isn't altered, but the epigenetic markers can persist through cell division, meaning that the corrected expression patterns are inherited by the daughter cells. Alternatively, if CRISPR technology is used to cut and insert a section of DNA, permanent changes occur in the DNA sequences, which are passed on through the new cells. Multi-generational impact. Inherited epigenetic modifications have the potential to impact the offspring of the individual receiving the therapy. This is because the epigenetic modifications may be transmitted through germline cells, which are egg or sperm cells, meaning that the offspring can inherit the altered gene expression pattern. This approach raises many ethical concerns, however, such as the fact that the technology will be used for enhancement rather than therapeutic reasons, which will be discussed later on, I believe. Its use is therefore restricted. Efficiency and cost effectiveness. CRISPR technology is faster and more reliable than previous gene editing techniques, such as TALENs and zinc finger nucleases, and allows for multiple genes to be edited simultaneously. It does not require complex protein engineering and can be learned relatively quickly by trained individuals. It is also cost-effective in some cases, meaning that smaller laboratories can perform advanced experiments. This efficient, cost-effective, and easy-to-use nature of CRISPR in comparison to previous technologies means that it may be accessible to many laboratories in the future, which can lead to faster advancements in the technology, faster results, and potentially greater accessibility for patients in the future. Thank you.
So the challenges, limitations, and ethics of CRISPR technology. So one of the main challenges is establishing a strong ethical framework. As CRISPR technology is starting to boom and moving at a faster pace in our scientific community, there are certain ethical issues that arise, and hence a strong ethical framework needs to be established. Previous examples, such as induced pluripotent stem cells and using embryonic stem cells in research, shows a very good example of why a strong ethical framework is required. The four main pillars of medical ethics, beneficence, non-maleficence, autonomy, and justice, all explore certain aspects of things that we need to consider as we establish CRISPR technology. So beneficence, we need to ensure that we promote well-being and health. For non-maleficence, we need to kind of avoid harm through its own consequences, which I'm going to elaborate further on. And autonomy, we need to kind of consider that future individuals cannot consent to treatments. And justice, as Shreya mentioned, we need to ensure that there is fair access and no socioeconomic disparities, because at the moment, treatment is quite expensive. And again, when we look at gene enhancement, which is one of the CRISPR technology types, we need to examine the nature of it. Is it for the sake of tackling or preventing disease? Or are we just manipulating phenotypes? And this is where the ethical framework comes in, because it prevents malicious intent coming in and ruling over decision making. And then designer babies. This is a famous term, and this is pretty much a term that implies that we manipulate the phenotypes and genotypes of babies. And again, there's a lot of ethical issues associated with this, and this is really important to mitigate through a strong ethical framework. And then next, when we look at CRISPR technology, yes, we're reducing disease, but at what cost? And as explained earlier, CRISPR technology has great potential to tackle diseases deemed incurable. Examples like cystic fibrosis, Huntington's disease, muscular dystrophy, and even more. However, this has unintended consequences. for instance, off-target effects. So off-target effects is pretty much a term to describe that this is when editing is happening on other DNA sites that is not really tackling your target sites. And then chromosomal translocation. Chromosomal translocation is when a section of a chromosome moves or becomes located to a different area. And this can activate oncogenic genes, which can potentially cause cancer. And this can also cause other problems as well. And this links to cellular stress and senescence. So if the DNA breaks during CRISPR technology gene editing, this can cause damage and change to how the DNA is transcribed. And if the damage is far too great, then this can cause the cell to stop dividing overall, which then leads to senescence and eventually aging and tissue dysfunction, hence cellular stress. And then immunogenicity. So immunogenicity is a very fancy term which just refers to the ability of a substance to trigger an immune response in the body. Sometimes the CRISPR components can be recognized as foreign, and this can cause the immune system to attack its own cells or reduce the effectiveness of the treatment, and can also cause inflammation or other adverse effects. And then the last point is biodiversity, which is both a limitation and a challenge. So biodiversity can be caused by the effects of gene enhancement. So because certain phenotypes and genotypes are chosen and preferred, a loss of genetic variation may occur. And this is because certain traits might be preferred, such as height, eye color. In fact, one study found that people that tend to have more favorable physical traits tend to earn more with their salary. So again, there's an ethical issue here associated. And then this can cause the gene pool to narrow over generations as less variation is within that particular pool. And then this biodiversity this doesn't really apply to only humans, but again, as mentioned earlier, other organisms too, such as plants and animals and insects, because CRISPR technology has been used in animals. An example of this is mosquitoes. And if this impacts the gene pool, then this will reduce ecosystem biodiversity and propose a new threat, as if selection pressures come in, for example, disease, then will they be able to withstand that? And again, this brings a question: what extent should humans control traits? So as you can see, biodiversity, the ethical framework, and the consequences of trying to tackle disease are all interlinked. But thank you, I'll now pass on.
So now I'm going to talk to you about a few case studies, specifically two that are really important in CRISPR history. The first one of these is editing blood, so CRISPR versus sickle cell disease. For those of you who don't know what sickle cell disease is, it's basically the name for a group of inherited health conditions that affect the red blood cells. The most serious type of this is called sickle cell anemia, and people with sickle cell disease produce unusually shaped red blood cells that can cause problems because they don't live as long as healthy blood cells do and can end up blocking normal blood vessels. So this case study is about Victoria Gray's breakthrough treatment. So in 2019, Victoria Gray became the first ever patient in the United States to receive CRISPR gene therapy for sickle cell disease, marking a really important moment for genetic medicine. The blood stem cells extracted from her were genetically modified using CRISPR-Cas9 technology, and these edited cells were programmed to produce fetal hemoglobin, which bypassed defective adult hemoglobin. After this treatment, she experienced a dramatic reduction in painful vaso-occlusive crisis, which basically means sickle cell crisis, where the blood cells are blocked and it causes excruciating pain for sickle cell patients. And these went from frequent episodes to virtually no episodes. The patient now lives independently without regular hospital visits for pain management, which is something sickle cell patients normally have to do. And this creates potential cure rather than symptom management for genetic blood disorders and provides hope to millions of people worldwide suffering from blood disorders. The next case is about He Jiankhu's CRISPR babies from 2018. So, for a background, in 2018, a Chinese scientist, He Jiankhu, announced the birth of twin girls whose embryos were edited using CRISPR to disable CCR5, which aimed for HIV resistance. The issue with this, however, was that the girls were not at direct risk from HIV. The edits may also not have worked as intended, which could have caused mosaicism, which is when the patient has two or more cell populations with different genetic or chromosomal compositions, and they were also conducted without proper ethical oversight or transparency. The outcome of this was that it sparked global outrage, and he was sentenced to prison in China. And this is really significant because it led to calls for stronger international regulation and moratoria on germline editing, which basically means that stronger regulations on the editing of the germline, which is inherited genetics. And it reinforced a clear boundary between the somatic and heritable genome editing. So now, as for all technology, that we have also considered new developments for CRISPR. So the first one is Cas12a's multiple gene system. So simultaneous editing of multiple genes in mouse models revealed complex immune interactions and disease mechanisms, which were previously impossible to study. This meant that doctors could study these mechanisms and provide potential cures, whereas previously we had no way of looking at these in deeper detail. The second development was base and prime editors. These are ultra-precise single-base changes without double-strand breaks and dramatically reduced target effects and improved safety profiles. It meant that the process was a lot safer for the patients. Another development is AI-guided designs, where foundation models integrated chromatin data to predict guide RNA activity and optimized efficiency whilst minimizing unintended edits, which also made it safer for the patient to experience. So from another clinical breakthrough would be from lab to life. So instead of just being an experiment, it actually happened. So Case GEY success stories relates to Victoria Gray's story, where the world's first CRISPR therapy demonstrated 96.6 success rate in preventing sickle cell crisis and is now available on the NHS, which wouldn't have been possible if we hadn't tried it out on people. There are also single dose miracles of different drugs. So NTLA 2001, 90% reduction in toxic proteins for hereditary amyloidosis. And there's also another one for Verb 101 with permanent cholesterol reduction with just one treatment, and YOL 203 with 70% reduction in urinary oxalate for rare kidney diseases. And these represent a shift from lifelong treatment to potential one-time cures. So instead of the patient going nearly every day or every week and having to spend their life surrounding this one disease, they can instead go for one treatment, which improves their quality of life. And for the further on in the future, scientists are thinking of cancer immunotherapy. So CRISPR-enhanced CAR T cells with PD-1 knockouts showed improved anti-tumor activity and potential for off-the-shelf treatment. So again, not needing loads and loads of time spent on one treatment, patients can just go in for a few or even just one treatment. and have the disease over and done with. There's also an autoimmune reset, which basically trials targeting B cells in lupus and other autoimmune conditions. And this aims to reset dysfunctional immune responses. So things like arthritis and anywhere where your immune system attacks you, they basically reset that function and make sure that it's working in your favor. There are also, on the side of plants, there are also climate resilient crops. So agricultural applications develop drought resistant, nutrient efficient crops to address global food security challenges, because as we know, the world's population is increasing and having more food is always better. So challenges and tomorrow's promise. So for personalized medicine, therapies have been created in 6 months for individual patients, and this truly improved personalized medicine. But there are also regulatory and cost challenges remaining, because personalizing medicine for each individual patient means that the medicine is also more expensive to make and trial. There are also delivery innovations, because we can now use lipid nanoparticles and improved viral vectors to expand the reach of CRISPR, whereas before there were some tissues that just couldn't be reached, including the brain and lungs. They were either unreachable or too sensitive. There are also ethical integration now, so balancing therapeutic potential with responsible oversight, and ensures equitable access while maintaining safety standards. So make sure that everyone can access it at the same quality instead of having it at worse quality for cheaper.
Thank you so much for coming, everyone. I will send this form in the chat, so please stay on the line for a few more minutes while I send it, and then after that you can leave.